There is a stark and almost disturbing contrast between the high-tech equipment used by these health workers and the rural African setting of those they have come to protect from the ravages of the rare but deadly Marburg virus.
Over the past few months, I've done my biotech project studying the Marburg virus. Marburg is classed as a filovirus, which means it comes from similar origins as Ebola. This made me wonder if Marburg could pose a similar threat as the Ebola virus has in the past. Right then, I decided I would find out. I narrowed down to this topic because when there was the Ebola outbreak in 2014, I was intrigued on how a virus could spread so quickly and affect so many people. When searching for topics this year for the project, this came to mind. I was set to do my project on Ebola originally, but then I discovered a lesser-known virus, Marburg virus, during my findings. Marburg seems to possess a similar structure of those in other immunosuppressive viruses, such as mumps and measles. The thing that makes it stand out to me is the dominant carrier of the virus is bats, primates, mice, and guinea pigs. In humans, less so. This is because this virus has some differing mechanisms in some of its viral proteins that are unique among other filoviruses. I thought that this shouldn't cause concern, but this virus can be just as devastating to a human system as Ebola can. There is no cure, and treatment options are very limited. Throughout this video, I examine some of Marburg's capabilities, and I compare and contrast the similarities and differences between Marburg and Ebola. I hope you find value in what I have researched over the past few months, and I hope you enjoy the show. The array of proteins on the virus help to revolve the cycle of creating, packaging, and spreading. The exterior receptors of the Marburg virus are called the glycoprotein. Glycoprotein is a carbon membrane bonded to the protein of the virus. Its responsibility is to communicate with other exterior host cells. GP is inserted into the host membrane as protein, which is composed of 3 identical units of polypeptide. This is known as a homotrimer. The processes of replication and transcription within the Marburg virus are dependent on nucleoprotein. Nucleoprotein forms its nucleocapsid shell on the virus by VP24, which combines nucleotides and the original outer protein called capsids. When NP or nuclear protein is expressed in the absence of other nucleocapsid proteins, it self-assembles into a highly organized tubular structure that resembles the nucleocapsids in its infected cells, indicating that it is the driving force for the nucleocapsid formation. NP serves as the viral hub for all the Marburg's viral proteins. A central coiled protein in the central part of NP has been shown to play an important role for self-assembly and budding, also for interacting and communicating with NP and VP35. GP contains a single open reading frame used for encoding the full length of the afflicted cell. This is used to translate the code from the other cell. Viral protein 24 is a protein that is important for the release of viral particles in the context of infection, although it influences neither the morphology of VP40's particles nor the efficiency of the particles' release. VP24 is unique compared to some of the other filoviruses because of its non-immunosuppressive nature. There is a weaker connection between the nuclear protein and VP24. Transcription factor VP30 is also involved in the replication of the Marburg virus. This protein contains positive sense RNA, or otherwise known as a start-stop signal. This process is activated by VP35. Once this process is started, the RNA combines with the DNA to create the viral DNA within the virus. This begins the process of spreading the disease. Finally, polymers, otherwise known as just L, is a protein that is multifunctional. It is dependent on the RNA and is used for catalytic functions for the virus. L contains the enzymic functions for the polymers and is believed to carry out RNA synthesis for viral DNA. One of the many ways the Marburg virus spreads its wrath from host cell to host cell is through a cycle known as the lytic cycle. The cycle begins when the viral DNA that is formed from the VP30 and VP35 is assembled into what's known as a DNA phage. DNA phages utilize viral DNA as well as protein from the manufacturer VPs. A DNA phage is released via VP30 and attaches itself to the cell within a host. Then, the phage transcribes the information to the cell, taking over its organelles to be used for synthesis of viral genomes and proteins. From this synthesis, more DNA phages begin to form, and then they all burst out, killing the cell. Once the phages are free, they seek out other cells in the host. The cycle continues to spiral until most or all of the cells are dead, resulting in the likely death of the host organism. A similar cycle to that of the lytic cycle is known as the lysogenic cycle. Instead of the outright attack of the host organelles on impact, this method involves the DNA phage to cripple the host cell over time. The phage penetrates the host cell just the same, but instead of releasing the viral DNA to corrupt the organelles, it molds with the genome of the cell. Later, the host cell will go through mitosis and split, but the virus still will be attached, making two copies. Eventually, one of the host cells will try repairing itself by using restriction enzymes to remove the harmful bacteria from the cell. At this point, the virus will begin attacking the cell, hijacking its organelles just like the lytic cycle. This then will produce DNA phages that will burst out of the cell, repeating the cycle once again. Some of the symptoms of the Marburg virus help show the resemblance of Ebola. Internal bleeding and coughing blood are common signs of the crippling disease. This is from the virus attacking your internal organs, especially your liver. From the lytic and lysogenic cycles, the organs might shut down from the host cell organelles being destroyed. Marburg also utilizes what's known as an acetylglycoprotein receptor. This serves as an attachment point to link the glycoprotein of the virus to the liver to kill the unwanted cells. This makes the process of cell elimination more convenient for the cell because of the direct contact to the liver from this receptor. From this, I can reason that the Marburg virus has similar awful effects compared to the Ebola virus and should definitely be concerning if you or someone you know has the virus. Little counteraction is known, and there is no solid cure. The best counteraction if you are infected is to make sure you drink lots of water, bleach your clothes, get plenty of sleep, and isolate yourself from the rest of the world. Now, what have studies of the Marburg virus shown us? The 1967 outbreak in Germany was caused by scientists relocating African green monkeys to study their tissues. These monkeys were documented to have been bitten by the African fruit bat, who is the host of the Marburg virus. Although this outbreak caused the death of 31 people, this sparked the interest of scientists to learn how quickly the Marburg virus can kill an organism. They studied the monkeys affected by the virus and concluded the average survival duration was between 6 and 13 days. Once deceased, scientists discovered next to none lymphocyte white blood cells left inside the primate's bloodstream, as well as significant weight loss. Numerous outbreaks of Marburg and Ebola in the primate community caused a great decline in their population. Ecological data suggests that it has contributed to an 80% decline in the local great ape populations in Gabon and DRC. Scientists also tested Marburg using aerosol transmission on animals, such as pigs and hamsters, getting similar results as the primates. This virus might not pose an enormous threat to us, but it might to other species. If a heavy outbreak were to occur in African wilderness areas, this could ignite the potential of many animal populations being reduced to the point of endangerment or extinction. On the basis of my research, the Marburg virus has the potential to pose a threat, but is not likely going to pose one. Even though the virus is rare, it has the dormant characteristics to strike in large quantities, killing up to hundreds or thousands. What should be taken away from my research is that a cure should be developed within the next few years to protect ourselves from a potential outbreak. Marburg could take us by surprise if we aren't prepared to handle the power this virus has. We don't have to repeat history from 2014. We can find a cure for it and hopefully eradicate the virus in its entirety. I hope this video gave you some insight on the true threat of the disease you probably haven't even heard of.